Hi, everybody. Hope everybody is safe and well and enjoying the conference. My name is Mark Daly, and I've been with Dovetail for actually going on 6 years now, almost. And I'm here with my colleague Mahal, who will be tag teaming this presentation. She's going to do uh, the second half. I'm gonna speak now for about 20 minutes. Here's the agenda. I'm going to talk about how we build genomes at Dovetail. And then I'm going to do something a little different, talking about challenging projects that we've encountered to give you a flavor for, projects that really push the limits of our technology and we're still successful with. And then I'll switch over to Mihal, who will talk about our latest advances in phasing and building true diploid assemblies. So how do we build genomes at Dovetail? Some of you may already be familiar with Dovetail, but it's probably still wise to listen to this because there are some advances we're making. We're always trying to optimize our workflow and technology. So this schematic here shows you the stepwise process for building genomes at Dovetail. The top lists the methods we use, and the bottom shows any kind of QC methodology that we employ to ensure we're delivering the best quality assemblies. So we first build a draft assembly using PacBio HiFi, typically around 40x coverage. We assemble with HiFi ASM. Again, here are some of the basic metrics we use to monitor the quality at this stage. After this point, we have a very high quality assembly at the base pair level, but it's in thousands of pieces. So we need a way to then put those thousands of contigs into chromosomes to make it useful for you. So for that, we use a step 2, which is scaffolding. The technology we employ for this is called OmniC. And I'm going to describe exactly how OmniC works in a second. We sequence these libraries on Illumina. Again, there's a variety of QC metrics we employ at this point. For contiguity, we look at the scaffold N50. For completeness, we typically look at BUSCO scores. After this point here, not only is your assembly going to be highly accurate, but also highly contiguous, typically chromosome scale. Step 3, annotation. So we can run RNA or iso-seq here and do a full annotation pipeline so that your genome is ready to publish. Just a quick backgrounder, because not everyone on the call may know how proximity ligation works. So I just want to make sure everyone's on the same playing field here. Essentially, what proximity ligation is, you might know this as Hi-C, but we now call our version of this OmniC to sort of differentiate it. But what happens with Hi-C or OmniC is in the nuclei, we actually fix the chromosomes with formalin. We then chop up the chromatin with an enzyme, allow everything to ligate back together, all those free ends. And the closer to any 2 points are in physical space, the more likely they are to ligate than 2 points that are farther away, as shown in this image. These points in the circle are more likely to ligate than these 2 points that are farther away. So what you get is this distance curve. The x-axis is linear distance or separation between the left and right reads, and the y-axis is probability. And so as you go down this curve, these are the interactions that are very close together. And as we go down the curve, the ligation interactions get farther and farther apart until we're literally at the telomeres. This little cartoon on the bottom just shows you our progression in technologies. We started the company in 2013 with Chicago, which was an in vitro form of this technology. We then released our restriction enzyme-based Hi-C in 2015, and OmniC was released a couple of years ago. And the distinct difference with OmniC is that, unlike traditional Hi-C, that is typically used by essentially everybody except Dovetail. We use DNase 1 to chop up the chromatin. And the problem with using a restriction enzyme, not a problem, but I should say sort of a challenge, is that it will only cut at certain sites, right? There's a restriction site that's specific for that enzyme. And if there are regions of the genome that lack that site, you're not going to get representation of that part of the genome. OmniC, on the other hand, uses DNase 1, which, as you know, does not cut at a certain sequence. It cuts anywhere. And so what that translates into is very even coverage of the entire genome. And of course, with any genetics or genomics experiments, you want to minimize, if not eliminate, bias in any way. So we've done that now with OmniC. Just very quickly, at the top is the molecular biology. You have intact chromatin in the nucleus. You cross-link it, chop it up with DNase 1, allow the free ends to ligate randomly, reverse the cross-links and sequence on Illumina. And we can use Illumina here because all we're interested in is 400 or 500 base pairs of these junctions. The dry lab, I'm going to switch to laser pointer here. The dry lab is shown on the bottom. This is our software called HiRise. And essentially what HiRise's job is to take the PacBio contigs, those tens of thousands of jigsaw puzzle pieces, and essentially take those with the proximity ligation data. And the proximity ligation data will help to determine the proper order and orientation of all those PacBio contigs. And it will basically create chromosomes from all those thousands of pieces of DNA. And so this is the schematic of HiRise, but essentially what comes out at the end is a very highly contiguous assembly, and it's essentially ready to annotate. Just as a reminder, I realize that a lot of the consortia are probably more interested in using kits in their own lab. And all that I've talked about now can, on the scaffolding side, can be easily represented in your lab using our kit, our OmniC kit. But if you do wish to, do a pilot or outsource some of the work to Dovetail, we do have the full service. Okay, so I want to prove to you, give you an idea of what the data actually looks like from OmniC. So I've said that the coverage is a lot more even. I want to actually show you what that looks like with real data. So here we have three different proximity ligation technologies: OmniC, single enzyme Hi-C, and multi-enzyme Hi-C. With single enzyme Hi-C, this is a sequence coverage plot. The ideal curve is shown in the dotted line. This would be representative of a shotgun library. This is the ideal. And you can see that the single enzyme Hi-C falls far short of the ideal because you have quite a bit of the genome that's either underrepresented or not represented at all. You also have quite a bit of the genome that is overrepresented. And this may not be so bad bioinformatically, but there's not really too much of a penalty for having that, but it's just a waste of sequencing resources. So what that looks like is a very bumpy coverage if you look at the raw data. What some people try and do to eliminate the bias or reduce it is add in a cocktail of different restriction enzymes. That helps the problem. As you can see, the curve is now more of a rounded curve, but it's still, you still have this problem here, right? And so when you look at the raw data, you're still getting this lots of peaks and valleys. When you switch to DNase 1, you get a curve very close to a shotgun library curve, translating into very even coverage. Yes, there are still a couple of valleys, but notice how the colored lines representing SNPs are all represented here. Most of the SNPs in this region of the genome are represented, whereas in the other Hi-C preps, There are SNPs completely missing, like these ones, and that is a problem for phasing and building diploid assemblies. And I'm going to let Mahal address that in her talk, but just to give you sort of a preview that it's really critical to represent as many SNPs as you possibly can in the proximity ligation data for purposes of phasing. Okay, I'm just going to quickly switch gears to challenging projects. Why do I want to talk about these? Luckily, we don't have a lot of challenging projects. Most of our projects, we've been doing this 8 years. We've done over 1,600 assemblies now, and most of our projects just sail right through the pipeline, no problem. But it's the challenging projects that I think really test the capabilities of a service provider. Those are the projects that really show whether you have the capability to do any kind of taxa that comes your way. And so that's why I really want to show you guys some examples of these. The 4 main challenges of genome assembly are shown on the left. So polyploidy, repeat content, high heterozygosity, and genome size. And all of these things get worse as they get bigger. So luckily, we're so experienced and we've got this so dialed in now. We very rarely hit upon projects that are what I would call ultra challenging. But again, those do exist. You're going into these assemblies blind and you don't always know how the genome or the sample is going to perform. But I listed these statistics here just to give you a feeling for our level of experience in doing this. And I think our repeat, the number of people that come back to us is really telling. We've delivered so many genomes now. We've covered, a huge breadth of phyla and orders of eukaryotic taxa. We've also delivered lots of kits. So again, the thing to remember with our OmniSeq technology is we've distilled all that experience and knowledge into a box that you can employ in your own lab. So that kit that we provide is actually based on the experience of these over 1,600 assemblies that we've delivered. We've also delivered lots of TAD projects, and these are the topologically associated domains that exist in the data that we can provide you. You're free to Google our tree of life, which is available online. If you just Google dovetail tree of life, you'll find our tree of life, and that phylogenetic tree shows you a sampling of some of the projects we've done. Okay, the first challenging project I want to go over is there's actually 2. These are beautiful trees, the giant sequoia and coast redwood. These were projects, a very collaborative team effort between UC Davis, Johns Hopkins University, and Save the Redwoods League. And these are just incredibly iconic trees that exist in California, very limited range, as you can see here. They're both threatened. I'm sure you've heard about the preservation efforts of the largest sequoia in the world, the largest tree in the world, how they had to wrap the trunk in foil due to the wildfire. So we were really excited to be a part of these 2 projects. The giant sequoia was published. Just a couple of facts: it's the largest tree on Earth by wood volume, but sadly it's endangered and declining. The threats being primarily reproductive success being limited by fire suppression. It's actually good sometimes to have fires around these trees because the pine cones need to have heat in order for the seeds to be expelled. Of course, the usual suspects like bark beetles, drought, and climate change are also big problems here. For the giant sequoia genome, the challenge with this genome is simply genome size primarily. It's a diploid, but it's got an 8 gigabase genome. And the The team that we collaborated with gave us a pre-made assembly that was built using nanopore and Illumina. We scaffolded it with our older technology, Chicago and Dovetail Hi-C. It really tested the Dovetail workflow at that time, just based on the sheer size of the genome, and also the repeat content was close to 80%. So extremely high repeat content and large genome size. We got this incredibly high-end 50 though after scaffolding, the largest scaffold ever assembled at 985 megabases. This set the record at that time. And now they have this really highly contiguous assembly. They're going to use it for a variety of genetic applications. The Coast Redwood, again, this is now the tallest tree on Earth. So it's not the largest by volume, but it's definitely the tallest. And the threats, again, primarily climate change, but logging, disease and pests, et cetera. Fire suppression is still there, just like with Sequoia. And a high quality assembly is critical to help protect this species. So very quickly here, the assembly, in this case, they used a similar strategy and gave us an assembly that we scaffolded using Chicago and Dovetail Hi-C. In this case, the genome is even bigger at 27 gigabases. And again, with very high repeat content. It's actually a hexaploid, but we were lucky in that nature gave us a loophole here because the actual pine nuts are haploid. So by using the genome of the pine nut, we were able to get around the polyploidy. I don't know if this would have even been possible had that haploid megagametophyte been available. Now we're setting a new record, largest scaffold ever at 1.3 gigabases. And again, lots of applications for this genome assembly that the team is employing. Moving on to marine invertebrates, these are very challenging creatures due to the way they reproduce. So this was a burrowing urchin project we did with Adam Rietzel and Remy Ketchum, University of North Carolina. They were both Matching Funds Award recipients for this project. This is a very interesting organism because it's the Persian Arabian Gulf has the warmest ocean temperatures on the planet, but it's very understudied in terms of how these creatures are adapting to extremes in temperature. So again, a very interesting project with what's happening with the warming oceans. So again, these creatures use external fertilization, which means they just spray their gametes into the water. That's a big problem for genome assembly because you have extremely high heterozygosity. So the first attempt at this, I believe that Adam and Remy used Illumina only. This was years ago, right? So they used Illumina only and weren't able to do an assembly. We then tried 10x Genomics to build the initial draft assembly. At that time, 10x was still able to do de novo assembly. They're not able to do it anymore, but we built an assembly. The problem was the assembly was twice the size we expected. And that's what you expect if the assembler is not able to do an assembly properly because of the heterozygosity. So the genome size is 900 megabases, and what it was doing was appending those two genomes to build a super genome of double the size, which obviously is not representative of the actual genome. So that was a problem. We tried scaffolding it anyways, but we had to then switch to PacBio. And when we switched to PacBio, we got the correct genome size, a nice N50 that we were able to then scaffold with Dovetail, Chicago and Hi-C. And we delivered a nice assembly at the end. The BOOSCO scores are nice and high, so we know the assembly is complete. And the genome size is correct, and the data showed chromosome scale assembly. The brilliant thigh poison frog, this is a project we did with Lauren O'Connell from Stanford. The challenge with this project is, again, large genome size, high repeat content. So there's the male, there's the frog there showing off his brilliant thighs. As you can see, it's a gorgeous little animal. Lauren was also a recipient of a Matching Funds Award. She's studying this critter for a variety of reasons, mainly regarding behaviors. So these guys, when you put them away from their nesting site, they can come back. They have a way of homing back to their home territory, which is very interesting. The dads carry the tadpoles on their back, which is also really an interesting behavior, and they're extremely territorial. So we built this 5 gigabase genome with 70% repeat content using Chicago and Hi-C, and unfortunately it failed. There were a couple of reasons why, due to the library construction, that it wasn't working. So luckily we had OmniC available at that time, and we switched to OmniC and got a successful assembly. So again, this shows that we have this passion for not quitting, and when we're working with these challenging assemblies. And you can see here that, again, the statistics for a genome like this are really good. You know, the Busco stats are in the mid 80s. The contiguity is high. So Lauren was really happy with this final assembly. So that is my time up. And I'm going to now hand this off to Mahal, who's going to talk to us about building chromosome scale haplotype resolved assemblies, and actually talking about our advances in diploid assembly, which are really exciting.
Hi, my name is Michal. I'm from Dovetail. Thank you, Mark, for the introduction. I will talk with you today on some very exciting development and how we can generate beautiful phased assembly, chromosome scale phased assemblies using OmniC and CCS reads. So CCS reads or hi-fi reads, PacBio reads, I'm sure the audience is familiar with. Just a very quick reminder, the idea is that we have the very long reads with N50 usually around 12 kilobase pairs or 15 kilobase pairs. And since we read the same molecule multiple times in a circular manner, a consensus call of the read can be built, generating hi-fi reads with a very high accuracy, similar to shotgun data, but with a much larger read length. That's one key technology that we are using, and the other one is OmniC that Mark already gave a very good introduction. So the main thing that I just want you to remember when we talk about OmniC is, in a way we have here when we use hi-fi and OmniC, it's not that we have 2 data sets, we can actually have 3 data sets, because we have the hi-fi reads, and the OmniC can serve both as the proximity ligation with a long-range linkage information, but also as a shotgun data. And in my next slide, I will show you how, before that, I will review on how a phase assembly project, what are the main steps or main component. First of all, we need to know what is the genome size. So genome size estimation that will dictate how much sequencing we need to do. Then we need to have the actual sequence in order to build the assembly, the de novo assembly. Next, usually this will yield contigs that are, or will always, they are not going to be chromosome scale. So we need additional information for scaffolding the contigs. We also want to be able to phase between the 2 copies. We're talking about diploid assemblies. And finally, it is always important to know the quality of the assembly. So contiguity, looking on N50, For example, what the largest scaffolds, that's one thing. Busco was also mentioned by Mark, looking on a set of genes that we expect to have in one copy, and that are very similar across different organisms. But that's not enough. So if we have a reference, a good quality reference, that's one way we can assess. But in the large majority of de novo assemblies, we don't have. These are non-model organisms, and we don't have a reference for evaluation. So we need to have some kind of reference-free way to evaluate the quality of the assembly. One way is to look on k-mer completeness, that I will talk about it later. And we also want to evaluate how accurate is the assembly. Do we have any errors that we introduced into the assembly? The first thing that we will cover is how similar are the OmniC reads into shotgun data, and can we use k-mer analysis with the OmniC reads? Now, k-mer analysis, it only means that you take substring of your sequence and you build a database of all the k-mers or of the substring that you decide that you have in your sequence. You can use, for example, 21 mers or 31 mers, and you see how bounded these k-mers in your data set. This example is from HC002. This is a genome in a battle sample, so it's from a Ashkenazi trio that is well studied, and this is shotgun data, and we can see here that the k-mer distribution is very typical. We see that the middle here is around 70, so that means that the coverage is around 70. Most of the k-mers, these are their coverage in our data set of the shotgun reads, and we have here this peak, which is around 35x, so about half of it. These are k-mers that are represented only once in our data set. So the main peak corresponds mainly to homozygous k-mers, so we will have 2 copies of them, and then you have a bunch of k-mers that have only 1 copies. These are the heterozygous sites, and this is typically some error sequencing. This is what we get when we look on shotgun data, and when we use the same, when we analyze, we do the same analysis using OmniC reads, we just get the exact same spectrum. This is not a copy paste. You can see here some minor differences, but that's really confident that we can use OmniC reads for k-mer analysis, which is very important in the assembly wall. And we start with the first project. This is a lizard de novo assembly project with in collaborations with Professor Dolby from Arizona State University. And what you see here is the k-mer distribution from OmniC reads, and using GenomeScope we can use this distribution in order to calculate what is expected genome size, and the expected genome size is 1.9 gigabase pairs. Hi-Fi reads were generated to about 30x coverage from this expected genome size, and we assembled them using 2 different assemblers, Hi-Fi Assam and Peregrine. Both generated assembly in a size that is very close to the expected, so almost 1.9 gigabase pairs, with a N50 of around 20 megabase pairs, so it's still not chromosome scale. Hi-Fi Assam at some advantage over Peregrine, and from now on we will focus on Hi-Fi Assam assemblies. So now we are not talking yet about phasing. We will move into phasing. So showed you, if we're thinking about the full project of genome assembly, then building the de novo assembly is using CCS reads, CCS or Hi-Fi. Sometimes I will call them CCS, sometimes Hi-Fi. Genome size estimation can be achieved using OmniC. What's next? We want to know the quality of this initial assembly. So we said we can do BUSCO. The completeness in terms of BUSCO looks very good, 97, almost 98%. But we want to know more than that. The BUSCO, it looks on a sub of the assembly, not all the assembly. And we want to know overall if all the sequences that we expect to see really found. So we do a k-mer analysis. So now we compare the k-mers that we found in the assemblies to the k-mers that we found in the OmniC reads. And we can see in this pink red region that most of the k-mers are shared between the lizard assembly and the reads. And here you see some region that is found in the reads, but not in the assembly itself. This will correspond to heterozygous site. Since we have this is not a phase assembly, we have only one copy. So some of the sites are just not in the assembly. And we also have this area where you find k-mers that are in the assembly and not in the reads. And that tells us about the errors that are generated due to consensus calls in the assembly. A great tool called Mercury allows us, using this information, to evaluate the completeness based on k-mers. So almost 98% completeness based on the k-mer analysis. So this is great. This is a reference-free quality evaluation. And the consensus quality value, which is 45.63. This is a logarithmic scale. So if you have Q30 will be in 99.9 accuracy at the base pair level, and at 40 we're already talking about 99.99. So the accuracy is very, very high. Okay, so we show that we can, besides genome size estimation, we can also use the OmniC reads in order to evaluate the quality of the assembly, k-mer completeness, and error rate. Next thing, we can also use, if we use, until now we use the shotgun-like data properties of the OmniC reads. Now we use the linkage, the proximity ligation properties of the OmniC reads. If this is before scaffolding, this is how the link density of the different contigs looks like. Okay, so, and then we use our high-rise software for scaffolding using the linkage information. And beautiful, we see this chromosome scale scaffolds. Since this is a lizard, you can see macro chromosomes and micro chromosomes and the micro chromosomes. And this is just a different way to visualize it. You can see the 8 largest scaffolds, or scaffolds that are bigger than 25 megabase pairs. And each black line represent a joining point between 2 different contigs. And you can see here all the stats with the largest scaffold now increasing after high rise from 86 megabase pairs to 380 megabase pairs, and overall N50 of 279. So OmniC reads can also, of course, help us for scaffolding. Next thing is phasing. Can OmniC reads help us do phasing? So we will review 2 different approaches for phasing using OmniC reads. One of them is k-mer-based approach. The other one will be SNP-based approach. We will start with a k-mer-based approach. So that this is a very recent publication, was published this month by Heng Li Group. It's a further development on the Hi-Fi Assam assembler. Now, Hi-Fi Assam generate graph-based assemblies, where you can see here, these are the different unitics. Okay, so these are contigs. These are contigs of the two different haplotypes in yellow and in blue. Green regions, these are regions with high homozygosity level, so there is no phasing available here. And typical graph assembly by Hi-Fi Assam will just have this, main structure here. But they are doing something very, very clever and using Hi-C to further support the phasing of the assemblies. And the idea is that you look for k-mer 31 mers in the assemblies that are unique. So only k-mers that are unique between the two areas in the graph are indexed, and then we map the Hi-C reads into this region and looking for Hi-C reads that have this unique k-mers, and using a fraction of these reads, only reads that have at least 2 unique k-mers. So it will help us bridge between regions that are haplotype specific. So since the Hi-C has a long range information, it can help us build those bridges between the different unitics. And one thing that is to remember here, even if Hi-Fi Assam won't be able to put these sequences together as a one complete sequence, like here we have all of this, there is some overlap, it can sometimes associate, it can know that this contig will be in association with these contigs, and it will bin it into 2 different haplotypes, and this is going to be important for the next steps. Okay, so I will try to go over it a bit more quickly. So if we are now looking on the lizard genome, we generated using Omni-C reads and CCS reads together, 2 different haplotypes. This is the previous one that we reviewed, and these are the 2 new haplotypes, and same genome size as expected, and we can see N50 after a high rise of around 280 megabase pairs. Again, completeness in Busco looks very good, and also when we look on the completer in terms of k-mer analysis and the consensus call is high. We can look on each one of the haplotypes together or calculate that for both haplotypes. So when we're looking on both haplotypes, 98% or almost 99% completeness. And we further check that we can use this approach also for more challenging assemblies. So sometimes plant genome assemblies are more complicated, they have higher repetitive content, but we can see here that this is for the brittle brush. We have 2 different haplotypes, each one of them is around 1.3 gigabase pairs, and 50 of 74 megabase pairs in both haplotypes, and amazing completeness. We are looking on the Busco. All the Busco genes were found in both haplotypes, and you can see how well it is scaffolded, each scaffold is well contained, the link density is well contained within the scaffold, and 99% completeness. Now, to be more sure about the results, it's always good to have some true set. So as we mentioned, we have the data set for AG002, where we generated Omni-C, in addition to the Hi-Fi reads that are available from the GIAB Genome in the Battle Consortium. And we generated de novo assembly using hi-fi SAM. We check the completeness. So now we have also the Illumina reads, but more than that, we have maternal and paternal, since this is a trio. And one thing that we notice that even if overall the completeness is 99%, by the way, it's very nice to see how very similar the results from the Omnisci and the Illumina are very similar. But we notice that one of the haplotypes have much less, much lower completeness versus the first haplotype. And if we look on the so we took these assemblies and we aligned them to AG38 and we got the explanation. So haplotype 2 just doesn't, since we have now a phase assembly, just doesn't have chromosome X. So this is, I didn't mention AG002 is this is a male, right? So we have only one copy of chromosome X. So that's already, okay, it looks very nice in terms of phasing. Now, since we have the paternal and the maternal information, the other thing that we can do is to look on k-mers that's specific to the maternal, to the maternal samples or to the paternal, and this is very beautiful. So each blob, each circle represent one contig from the assembly, and we can we check how many unique paternal k-mers that it says and how many maternals. And what we can see when it goes on the axis is it means that each contig is either just paternal sequence or maternal. If we had something in between, that would mean that you have in one context a mixture of k-mers from the paternal or maternal. But so that's really show that the phasing works amazing. But that's at the contig level. What happens when we assemble it? And the results are really amazing. So we have one haplotype which 3 gigabase pairs, the other one 2.88 gigabase pairs, and 50 around 145, which is exactly what you expect when you have the full genome, and max scaffold size, which is very close to the of 237, which is very close to chromosome 1. And you can see this beautiful link density. This is here the coverage is lower because this is chromosome X, where it's found in haplotype 1 and not haplotype 2. And we now look again on the same k-mer analysis with the maternal and the paternal information. And even after scaffolding, this is well maintained. So each scaffold, these 237 megabase pairs, have only or mainly maternal k-mers or mainly paternal. So the phasing works really, really beautiful. And now I will go quickly to the next approach. So SNP-based approach. So when we are looking, actually when we're trying to do phasing, we want to find variation between the two haplotypes in order to differentiate them. So k-mer is one approach, but another well-established way to find variation, genetic variation, is using SNP calling. So the basis for this approach is just doing a typical unphased assembly pipeline. You have unphased CCS reads and then unphased contigs, and you scaffold them with Omnisci reads. That's very typical. And then you take this assembly, and this will be the first step for the phased assembly. The nice thing about this approach is that it can work with your assembler of choice. So it doesn't need to be, it can be hi-fi SAM, it can be Peregrine, it can be something else. We take this unphased scaffold and we use the Omnisci reads in order to call SNPs. So we call SNPs. Deep variants seem to be the best caller from what we tested. And then we can phase the SNPs using HapCat. And we do the same also, and then we use WhatsApp for partitioning the CCS reads and the Omnisci reads based on this SNP calling. And now we have 2 different bins, one for each haplotype, of separate CCS reads and separate Omnisci reads. And for each one of them, we just regenerate, we take the bin CCS reads and we just build a new assembly. So 2 different assemblies. Okay, so this is just a review of that, of all the steps. So we want to make sure that that SNP calling is works well with OmniC, although we are already quite confident about that. So shotgun data is, again, it's the gold standard for SNP calling. In this case, this is with a GM12878 human sample, and we can see that we have, when we call SNPs using OmniC and shotgun, maybe in terms of sensitivity, OmniC is just a tiny bit lower than shotgun, but overall it is very similar. But we want also to be able to do phasing. So when we try, when we check the phasing using shotgun data versus OmniC, we can really see the strength of the OmniC type of data. With the shotgun data, you have very high error switch error rate for phasing and much lower with OmniC. Furthermore, with OmniC, you can have chromosome, you have all the 23 chromosomes phased end to end. This is by SNP calling, okay, so not assembly yet. With shotgun, of course, you don't have this this long range information, and none of the chromosomes have that. So what we wanted to see, we did this partitioning via using SNPs for the CCS reads, and we used our OmniC data, and we used also the trio information that I mentioned before, and 2.6 million CCS reads were assigned by trio to each one of the haplotypes, and similar with the OmniC data. When we look on the accuracy, so the agreement between the partitioning of the CCS reads in trio versus OmniC, there was almost 95% agreement, so the 97% accuracy. So with this approach, as I mentioned, we now generate 2 separate assemblies. Each one of them was around 2.8 gigabase pairs. In this case, chromosome X is in both assemblies, but we have the N50 as expected around 150, and the maximum scaffold size around 232 megabase pairs, very close to what we expect. What we can do with it, I'll go very quickly. We can do look on SNPs between the 2 different haplotypes. This is once we have the phase assembly. Skip that. The other thing that we can do now is look on the structural variation, which is something that usually is very difficult to see. So when we look on the 2 different haplotypes, we can see a 25 kb deletion in one of them and not in the other. In this case, we also had some other 10x linked reads, which also support this 25 kb deletion. So we can find structural variation between the 2 haplotypes. And we also show that this approach, we tested it also with the Atlantic bluefin. This is in collaboration with Professor Block from Stanford University. And we saw that we were able to generate 2 phased haplotypes with a majority of the sequence contained in the largest 24 scaffold, which is exactly what we expect. So just to summarize, I showed you that using OmniC and CCS reads, we can do all the steps of phase assembly projects, including genome size estimation, scaffolding, phasing, and k-mer completeness and error rate estimation. I just want to say a big thank you to, there were many people involved in this project, including collaboration with PacBio, Professor Block from Stanford, Professor Dolby from Arizona State Universities, and many people from Dovetail Genomics. Thank you very much.